In my prior video, I showed the synthesis of the RNA nucleotides: adenosine A, guanosine G, cytidine C, uridine U, and the DNA nucleotides deoxy-adenosine A, guanosine G, cytidine C, and thymidine T. So, how are these nucleotides assembled into RNA and DNA strands? Purines complement pyrimidines, which means the purine pairs with its complementary pyrimidine. So, the guanosine G pairs with the cytidine C in both DNA and RNA, and we see GC or CG pairs. In DNA, the adenosine A pairs with the thymidine T. In RNA, the adenosine A pairs with the uridine U, and we see AU pairs or UA pairs. It is the bases that do the pairing. The nucleobases guanine G pairs with cytosine C to form a GC pair. In DNA, the nucleobases adenine A pairs with thymine T to form an AT pair. In RNA, adenine A pairs with uracil U to form an AU pair. The bond between the bases are weak hydrogen bonds. There are 2 hydrogen bonds between the AT pair. There are 3 hydrogen bonds between the GC pair. So the AT bond is weaker, and the AT pair are more easily separated. Thus, the relative hydrogen bond strength can be important when it is time to separate the strands. It will require less energy to break the AT bonds. It is more likely the separation process will start at the weaker AT pairing. In assembling DNA, it is important to remember that the OH group of deoxy-ribose is on the 3' carbon, and the phosphate group is on the 5' carbon. The nucleobase is on the 1' carbon as shown in the figure. The DNA sugar phosphate backbone is assembled when the phosphate at the 5' carbon of the lower nucleotide joins at the OH group at the 3' carbon on the sugar part of the nucleotide above it. A water molecule, H2O, is kicked out during the bonding action. The bond between the nucleotides is a very strong covalent phosphodiester bond. The phosphodiester bond is catalyzed by a ribo-phosphate dehydrogenase enzyme. If we look at the fully assembled DNA structure, we can note the complementary pairs are matched in anti-parallel order. In the figures we see on the left DNA strand, the sugar phosphate backbone is arranged in the 5' to 3' carbon order, counting from the top. The right-hand DNA strand is arranged in an anti-parallel 5' to 3' carbon order, counting from the bottom. The anti-parallel order can be seen in the figures. In the figures on the left sugar backbone, the 5' carbon with the phosphate is pointed up, and the 3' carbon with the OH is pointed down. On the right sugar backbone, the sugars and the nucleotides are upside down, with the 5' carbon and phosphate pointing down, and the 3' carbon and OH pointing up. The nucleotides are anti-parallel to each other. The anti-parallel order will become important during RNA transcription or during DNA replication. The strand that is being copied is always read in the 3' to 5' carbon direction by the transcription enzyme. The new strand is always assembled in 5' onto 3' order, as shown in the figure. When constructing a new copy, there needs to be an OH group on the 3' carbon end for the incoming 5' phosphate to join and form the phosphodiester bond. I will talk more about transcription and replication later. As shown in the figure, the DNA is read from 3' to 5' on the red strand, and the copied strand in blue is synthesized in the same direction in anti-parallel order from 5' to 3'. The numbers are the carbon locations of the deoxy-ribose sugar. We can see in the figures that there are 2 hydrogen bonds between the AT pairings. There are 3 hydrogen bonds between the GC pairings. The figure shows the DNA structure is anti-parallel in the 5' to 3' carbon order on the left and right DNA strands. The DNA ladder twists into a double helix due to puckers or out-of-plane atoms on the sugar rings. The twist rate can be 8 to 10 nucleotides per twist. The figure shows the DNA structure and the anti-parallel 5' to 3' carbon order on the DNA strands. On the left sugar backbone, the 5' carbon with the phosphate is pointed up, and the 3' carbon with the OH is pointed down. On the right sugar backbone, the sugars and the nucleotides are upside down, with the 5' carbon and phosphate pointing down, and the 3' carbon and OH pointing up. The nucleotides are anti-parallel to each other. To summarize, we know that DNA is made up of the nucleotides adenosine A, guanosine G, cytidine C, and thymidine T. The nucleobases bond to each other in complementary purine to pyrimidine pairs. 2 hydrogen bonds form between the A-T pair. 3 hydrogen bonds form between the G-C pair. The sugar phosphates on the nucleotides form the twin backbones of the DNA molecule. The backbones are the result of phosphodiester bonds that form between the phosphate on the 5' carbon of the sugar ring and the OH on the 3' carbon of the sugar ring. The nucleotides are assembled in an antiparallel arrangement. 5' to 3' down one side and 3' to 5' down the parallel side. The out-of-plane carbon puckers in the sugar rings cause the DNA molecules to twist into a double helix with generally 8 to 10 nucleotides per twist. The nucleotides in DNA code for genes. A gene is a sequence of AGCT that code for one or more proteins. Most useful DNA sequences are interspersed with sequences of DNA code that, so far as we can tell, is nonsense sequences that have no useful function. Some coding sequences tell the polymerase enzymes where to read and transcribe the DNA and RNA, where a gene starts and where it ends. The most basic thing to remember is that strings of useful DNA code for amino acids. A codon is 3 letters of DNA that code for a specific amino acid. Enzymes read and transcribe the DNA sequence onto mRNA. The mRNA carries the message to a ribosome that assemble amino acids into proteins. For example, the RNA sequence GAU is the codon for the amino acid aspartate. When the ribosome that assembles the protein reads GAU in the mRNA code, it knows the next amino acid it must add to make the protein it is assembling is aspartate. Always remember the DNA-RNA protein cycle is life. At the molecular level, DNA codes for RNA. RNA codes for protein enzymes. The protein enzymes make more DNA. That is how life works.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.